So just to introduce you, Huw Evans, this part of the name is coming from Garfagnana area. So he's a Tuscanian, and so he will talk about remote monitoring and the detection of invasive species of hornets, Vespa velutina and Vespa mandarina, using acoustic tools. So you have 3 minutes.
3 minutes, that's all.
Okay. 10 minutes. Okay, thank you very much.
I'll go quick. Okay. Good afternoon. I hope you enjoyed your lunch. Beehero is a company based in California, in Silicon Valley. They predominantly focus on honeybees and selling pollination services to the almond industry of California. I head the innovation arm of Beehero, Beehero X. It's a bit like Google X, if anybody's heard of that. And I do the wacky stuff, like looking at monitoring bumblebees in polytunnels, looking at honeybees in crops for seed production and things like that. And also, of course, detecting Asian hornets. And what we're going to focus on in this presentation is trying to identify Asian hornets by the noise they make, which allows remote monitoring of these invasive species, creatures. So here's a great photograph of Asian hornets taken by Sandra, who's just sitting down in a seat there. I should also say that it was Sandra. What made this picture possible was Sandra, who introduced me to Antonio, who then introduced me to Fabrizio. So without these three cool people, this photo wouldn't exist and we couldn't have done the work we did. So here's Sandra taking more of that sort of type of photo. This is back in 2018, but you can see the little laptop in the grass here. And our aim at the time was to record the noises of the hornets. So there's me with a microphone in front of the hive, trying to listen to the noise of the hornets to see if we could find what we call an acoustic signature. It's a noise a hornet makes that isn't too easily confused with other noises in the environment around it. Just out of interest, that apiary about less than a kilometre away was a nest, which it's actually been destroyed here, but the hornets chose to make a nest in a tree outside the world's first 6-star hotel, which was named Fear. That is actually the surname of the gentleman that made the hotel. Then if we fast forward to 2020, the murder hornets, or Vespa mandarinia, arrived in Washington State in America. There was a lot of hoo-ha in the press about it. We got involved with the USDA. I should give a shout out to this gentleman because this was back in COVID times. And the first sound samples I saw from Vespa mandarinia were from Shuraki Mankio, who works for the Forestry and Forest Department of Japan. He sent me some video recordings with some recordings, but then very quickly we started working with Chris and Todd and Anne. A lot of people talking about Chris yesterday, seems to be the go-to guy. Here he is dressed as a spaceman and with his team extracting the Vespa mandarinia up in Washington State. And halfway up this tree, there was a nest, and it was going to take 3 days before they exterminated the nest. So I asked, could they put one of our acoustic monitors next to the entrance of the nest and allow us to record the noises of the mandarinia? We had lots of velutina recordings, but not so much of the, we had some velutina recordings, but not much mandarinia. So this monitor sat there for 3 days, there it is, and it got hundreds and hundreds of sound samples of hornets coming and going from the nest. So this is what a sound looks like, if that makes sense. This is what you call a spectrogram. So this is along a 16-second duration, and basically up and down is the frequencies. It's a stereo. This is if we take a section of this and look at the frequency components that make up that noise, we can see what we've got, what we call a fundamental frequency at 100 hertz, and then harmonics at 200, 300, and 400 hertz. Now, going through the sound files and seeing when we could see the hornets flying or not flying, we could actually map a daily flight profile of the hornets from the nest. So there's the 3 days. We could see the flight profile through those days. We could extract relatively useful information like the time they started flying. This gives you a slightly better idea of their behavior. Chris confirmed this is pretty much when they started flying. It all seemed to make sense. We didn't see any at night, obviously. You can extrapolate to how many hornets an hour are coming in and out the nest. It was about 400 hornets an hour. You kind of wonder if that's credible. It's like one hornet every 10 seconds. And if you can imagine looking at the nest, that kind of sounds about right. We also do a lot of monitoring of honeybee colonies. And when they're busy, about 6 bees a second are coming and going. So there's always a lot more activity than you think. So also Chris used this huge net and grabbed a couple of hornet samples and put them in this little mini flight tent with what we call our acoustic monitor. He calls that our soundometer. So we put a couple of different hornets in with our soundometer, and we looked at the audio files, and we could see there's a slightly different frequency. One of them was slightly lower pitched, one was slightly higher pitched. And we concluded this was due to a size difference in the hornets. So we got back to Chris with this, and he then emailed saying, yes, here, one of them was bigger, one of them was smaller. Here's the lengths and the weights, etc. And an important point here is that size matters, because the change in sound depends on the size of the creature. Now, this isn't the same with honeybees. Honeybees worker honeybees tend to be the same size. They don't really grow much once they've started flying. So they tend to be the same size, but they also differ in sound depending how much weight they're carrying in their stomach, whether it be nectar, not so much pollen because it's quite light, but certainly when they're full of nectar, they because of physics, they have to flap a bit hard and they change, but they all tend to be the same size. The hornets, on the other hand, hawking around in front of the hives, tend to have the same amount of stuff in their stomach. It's more the different size that gives a different sound or a different tone, different higher or lower pitched. So knowing the frequencies they generated and the dimensions and weights they were, we could do a very quick and dirty extrapolation of how frequency changes with size. And then we could look back at all the hornets we'd recorded coming and going from the nest, and we could work out the demographic of the nest, who was flying when. The smallest hornet would work out to be about 22 millimeters long, the largest just over 36 millimeters long. I'd be very surprised. It's only a 2-point graph, so I'd be very surprised if it was that linear, but it seems roughly reasonable. The idea is that we take these acoustic sensors up in the outbacks of Washington State. We put them around in the forest. These forests, it takes 3 days just to walk there, so it's a very, even with tracks, it takes a long time to go and check them. So the idea is, if we see a hornet, we can tell a communication gateway about that. There's no cell signal as there are no roads.
1 minute left.
Really?
Yeah.
So we can use satellite to send that back, send any alerts back of that. This is actually my hand. This is a little swarm satellite. It's the size of a slice of toast, but we can move that to that later. Fast forward to 2023, and the Vespa velutina arrived, as we've all heard. If we look at this US climate suitability, you can see that it mapped quite well to their abundance in Europe, or where they may go. And if you look at America first, you can see up here Washington State. But of course, this is where you wouldn't want to find a hornet, and where they may strive a lot more. And of course, that's exactly where they've just been found. So it has caused alarm. The USDA got in touch. So we took our little Beehero sensors and we went to an apiary mount suspender. So thanks to Stefano for this, and of course Antonio. And we recorded some Vespa velutinas. We used a little Beehero sensor. It usually lives in a beehive. It monitors humidities, temperatures, but it also listens to the heartbeat of what's going on in the hive to help the commercial beekeepers with their management practices and things. And we stuck some of these sensors on the hives at the Cooperativa Fiori, which we should be going to tomorrow on a field trip and see these sensors. I think their batteries are flat, but they did enable us to record lots of Vespa velutina noises. So looking, how does it differ? It's slightly higher pitched because the creature is slightly smaller. So physics would dictate it would be slightly higher pitched. It's about 20 hertz higher pitched, but small Vespa mandarinia get up there as well. So there's definitely an overlap there. During a field trip in Israel, I was lucky enough to find some orientalis. I didn't have the equipment with me, but when people say, what's the best microphone? It's the one you've got on you when you need it. So using my iPhone, I recorded the noises of the orientalis coming and going. And we can see they make the same noises pretty much as the mandarinia. Certainly not different enough to distinguish. So all the Asian hornets tend to make, well, of these three, similar sort of noise, different to bees. Here's one for Antonio. Crab rub. We also recorded some crab rub back in 2018 at the Pisa training apiary or something. Do you remember that afternoon we spent? And we recorded lots of them. And here's 3 examples. They're pretty similar to each other. But the good news is they're very different to the Asian hornet. Their peak is here, where the trough, if you like, of the Vespa velutina and the mandarinia and the orientalis are. So being able to distinguish between the crab roe and the Asian hornets is pretty straightforward. A quick thing on false positives: not only do you need to recognize the hornets, you need to repel things that aren't hornets and not set off too many false alarms. Traffic noise is good, barking dogs is good, hammer drills is good, Vespa scooters are good. Voice doesn't like voice. Voice can, we look for the sound every half second, continuously, 24/7. And there's a lot of parts of voice with people speaking that sound a bit like a hornet, and that can trigger it. Now, in the outback of Washington State, where there's no people, this isn't a problem. Even in an apiary, it's deserted and nothing but bees. We distinguish from bees very nicely. That's not a problem. But if beekeepers turn up and speak, it will trigger this algorithm. Any questions?
Perfectly in time with my compliments. We have more 5 minutes for questions. So
if the first question was, what's this data for? Beehero have no commercial interest in using this or charging anybody to monitor for hornets. This is all open source, if you like. You can record these noises on all sorts of things, and it's not a difficult thing to make an algorithm to recognize. I'm happy to share the algorithm and share all the data we have on it. It's not a, this isn't a pitch.
My first question was if the weight is important, then the sound is different when the hornet arrives to hunt and when it goes with a beak, or it is always the same?
No. If it's carrying, I mean, they chop off the abdomen, they chop off the head and the wings, and they just grab the thorax to fly away with. And this would make a huge difference to their sound. But they tend to grab the bee, like if the sensor's at the front of the hive, they grab the bee and they fall on the ground, and then they start decapitating it and taking the bits, and then they kind of fly off. So the sensor doesn't really hear the hornet carrying the thorax in front of the hive. But most noise they make anyway is just hawking, waiting to grab a bee. Ah, well, this is when that sensor's normally used. So that's a Beehero sensor, and they use them in commercial beehives for bee husbandry, just to see what a bee's doing. It's got nothing to do with hornets. That's just where the technology comes from. Oh, good on.
How would users of your acoustic detector be able to verify an alert? There are inevitably some false positives and false negatives with all automated detectors that, if not easily verified, can undermine confidence in the system. How would you overcome this with your systems? These are 3 questions.
Yes. Okay. Well, basically, the sensor itself decides if it thinks it's a hornet, but while it sends its alert, it'll send a message back to the server saying, okay, I think it's a hornet. Along with that, it sends all the data it used, it sends back all the data that the algorithm used to decide it was a hornet. So a human being can give it a sanity check. So as well as sending an alert, it'll say, here's an alert, and here's all the data that I thought I saw a hornet in.
Okay, so thank you very much.
Pleasure.
We can pass to the next speaker. And the next speaker is Pete Kennedy, and I'm very anxious to hear him because I'm trying to use his infernal machine. And of course, I lost 2 tags. So now I think that we tell us how we can find again our tags. So, okay, Pete, the connection is ready.
So I'll try and be quick, and because of that, I will skim a lot of this. So hopefully I'll be able to answer more specific questions in the question time at the end, hoping that there'll be time. So I'm focusing on some of the advances in the use of VHF radio tracking to locate Vespa velutina nests, essentially since our paper came out. So just to recap, back in 2017, so this was the year after the first Asian hornet nest, Vespa velutina nest, was discovered in the UK. The UK government actually approached us to explore to what extent sort of off-the-shelf technology could be adapted to help locate these nests. And the idea was very much to use what was already available rather than developing new technology, because there was an awareness that there's usually a long lead time in the development of new technology. So looking at what was currently available, we looked at various options, but to cut a long story short, we ended up focusing on radio telemetry as that we found was the most promising. And that work was then published in a paper which a number of people already referred to, so I'm sure most of the audience are familiar with. So that was published in 2018, Communications Biology. And since then, we have actually continued using radio telemetry, and we had the possibility under the Atlantic Positive project. One of the aspects that we did under this project was to further explore refinements in radio telemetry and also provide more comprehensive training materials to enable others to use radio telemetry. So really taking a step back, basically radio tracking is a well-proven technique in terms of tracking wildlife, but more commonly used for larger animals and has only recently been used for insects, largely to do with how the technology has moved on. It can be used to track individuals, but also individuals that belong to social groups and thereby be able to potentially follow those social groups, whether that is a pack of lions, a herd of goats, a flock of birds, etc. But it can equally be used to follow an individual to locate that individual's nest when that individual is part of a social grouping. The factors that limit radio tracking are obviously the size of the radio tags, their weight, their shape, and also in how you attach the radio tags. So these were things that we were keen to sort of overcome in our sort of early work on this. Unlike a lot of ecological behavioral studies with radio tracking, we weren't limited by a typical requirement of studying normal behavior, which usually means that you can't use a tag that's heavier than 5% of the body weight of the animal you're following. Thankfully, that's not the case. We don't need normal behavior. We only need the insect to be able to make it back to the nest, and therefore we're able to use these radio tags. The tags we mainly used were for the 2018 paper were Lotex, Picopip, AG337 and the AG190, which are sort of relatively large tags, but they were the smallest tags available at that point in time. And what has happened since that paper was published is that there's been development of new lighter tags and even tags that can now be recharged that have come onto the market. So examples of this are the Lotex Nanopin and the ATS T15 Tiny Tag. Both are about half the weight of the tags we were originally working with. And there's a similarly light tag, that's the Lowland Electronics one, which is a relative newcomer. And this one is one of the ones that can be recharged as well if you reacquire your tag. So this gives you an overview of the tags available from different manufacturers, with the ones towards the top being the heavier tags, the ones towards the bottom being the smaller tags. And to the right hand, on the right hand columns, you can see the sort of weights that of hornets that you'd be able to track with these tags. So you can clearly see that with the lighter tags, you can track smaller hornets than you would have been able to with the heavier tags. The paper often, people will often look at the paper and understand that hornets can carry up to 8% of their body weight, but ideally you're looking for a tag that's probably more around 65, 70% of the body weight to be really sure that it will be a good flyer. The 2018 paper, in that we anesthetized hornets to be able to handle them. We attached, we're able to manhandle them onto a sort of restraining plate. This then allowed us to attach the radio tags in relative safety. And as I mentioned, we provided generated detailed training material. There's a detailed manual available via this link. It also includes videos of particular aspects of the work. And since that method was published, an increasing number of people have actually been using radio tracking. People from Switzerland, Germany, Belgium, Netherlands, also in Spain, France, Channel Islands, and also in the USA. As Quentin was saying earlier, in Spain and France, because of the sheer number of nests, radio telemetry isn't really going to provide a solution there, other than perhaps in those regions where the hornets are only just arriving. It's really more of a technique meant to be used at the earlier stage of establishment, when it is vital you find ideally all nests, but it definitely as many nests as possible as quickly as you can. With a growing number of people who are using radio tracking, there's also a lot of experience being gained there are sort of continual refinements that I'm glad to say are being shared amongst practitioners, which allows me to actually share some of these here next. So there's two aspects. So first, with the lighter radio tags, because it's a smaller battery, that does then therefore mean that the signal that is produced is not as strong as with the stronger batteries, the larger tags. So you're typically talking about at least half the detection range of the larger tags. But that one also has to allow for the fact that in more complex environments, heavily vegetated environments, the detection range is reduced even further. So this can cause problems when a hornet flies off and then moves beyond the detection range. And this is where it's quite important to emphasize that the best practice is really to start with monitoring flight directions of hornets coming to a bait station first. You want hornets that are regular visitors, that aren't going to be frightened off by you trying to market the individuals. They are well accustomed to this bait location. You can therefore get the flight directions, record time intervals to get an idea of distance to the nest, and also you can weigh the promising individuals. And it's actually important to weigh these individuals when they arrive at a bait station, particularly at one of the sort of sweet bait, carbohydrate bait based baits, as you don't want them to fill their stomachs and therefore give you a false impression of weight. They can gain as much as 100 milligrams just by feeding on a syrup, etc. Once you know the approximate direction of the nest, even if you've got an idea of distance, you may even be able to find the nest without using radio telemetry. But the advantage here is that if you do need to use radio telemetry, you know the direction to continue in, even if the if the hornet flies beyond detection range. Another advantage with this is actually that as the hornets are used to returning to this bait, you have the opportunity to recapture an individual. After you found the nest, you can wait at the bait station for the tagged hornet to return, remove the tag, and therefore make it available for reuse, which is obviously beneficial in terms of costings. The other aspect I wanted to mention, it was something we were aware of earlier on, that cold anesthesia does have a sort of consequence on flight performance. There's an energetic cost sort of recovering from cold anesthesia, and obviously, so if it can be avoided, then it is beneficial. Although we were aware that many people would be quite uncomfortable sort of handling sort of live hornets. But as growing numbers of teams have been using radio tracking, more and more of them have been looking at skipping anesthesia. And I've come up with various ways of doing this. Important is that you avoid or minimize stress and damage to hornet in doing so, and also ensure the secure placement of the radio tag. And I'm just showing here, so pictorially, one example of how this might be done. So on the left-hand side, you have a radio tag that's pre-prepared via using a pipette tip with a loop sort of going through that. It basically allows me by pulling the thread, I can close the loop, and I can do this by sort of a little bit remotely from the hornet by using the pipette tip. I can catch your hornet, use a sort of double-pronged curling clip to hold the hornet sort of across the head and the thorax, invert the net to expose the abdomen, place the loop over the abdomen, sort of pushing it right down, sort of around the pedestal, and at that point, using the pipette tip and pulling on the thread to tighten the knot around the pedestal. A little bit of glue on the knot, if you can get it, is advised to just make sure that doesn't undo. So that's one relatively quick way of being able to attach a radio tag that then avoids the cold anesthesia. And that is particularly advised in areas where the climate might be a little bit colder, and therefore the hornets might suffer a little bit more or struggle a little bit more in terms of warming themselves up again. So that was just a quick overview. Really just like to sort of say thanks to the various radio tracking teams that have shared their experiences from Galicia, from Jersey, from Belgium, from Germany, from Switzerland, UK, Holland, and the USA, and also to thank my University of Exeter colleagues as well, who have helped with some of this work. That was a quick summary that hopefully allows time for questions.
Thank you very much, Pete. And perfectly in time, we have 4 minutes for questions. There are questions from the room? No question? It's quite hard to use this telemetry. I've tried it, so maybe it's a very important topic. And I thank again Pete because it's very illuminative for me, and it was really important. There is Federico Kappa that had a question, a short question, please, Federico.
Pete, sorry, have you tried to attach the tag by doing it on the thorax instead of the abdomen with the? Does it work?
So it does depend on what radio tag we're talking about. When I first started doing the work, I actually started by attaching them onto the thorax, but with the AG337, the AG190, the tags are essentially too heavy. So as soon as the hornet banks in its flight, it basically just falls over, drops to the ground. It's unable to fly. It changes its center of gravity too much. But with the smaller radio tags, I must confess I haven't actually tried it with those. Once I actually established that it works well by attaching the tag underneath the hornet, it's a lot easier for it to fly with a weight below it. It was in essence the method I used thereafter. It is possible, it might be possible with the lighter tags, but I haven't tried it.
And very quick, about the anesthesia, have you tried with CO2?
Very early on, I tried it with CO2, but I believe Sandra is in the audience as well. She also looked at CO2. One of the problems I found with CO2 is that it acts very quickly, but that also means that a millisecond longer of dosing has a potentially large impact. So the chances of overdosing or underdosing are a lot easier with CO2 than it is with using just sort of ice. Other teams I do know have been exploring CO2, but it's yeah, some success. But I think, as has been alluded by Antonio, people are still finding aspects of the work, there are nuances to it that are a little bit tricky. And in some ways, rather than adding sort of additional nuances, I'm trying to simplify it to make it as sort of ideally as standardized for everybody.
So thank you very much, Pete. We can pass to directly Laura Bortolotti. Laura Bortolotti will give a talk on the first results of the Z method for the indirect neutralization of nests.
So I will present the first results of this method Z. What is the method Z? Z is for Zani, our colleague here, who invented this method, who allowed the neutralization of nests indirectly to the contamination of hornets. So is it a Trojan? Yes, it is. So I know that is quite controversial, and some colleagues here gave their opinion on that. But what I want to point out is that it's not a poisoned bait, it's a direct contamination, so it's 100% selective. The dosage is a micro dosage, so very little amount of active ingredient. And we run this test in a research project under authority control. And we awaited the official authorization from the Italian Ministry of Health in 2 regions of Italy. The most infested one in region in Liguria region. Of course, following all this procedure, there is also a risk assessment procedure, so it's not left on the hand of everybody. We started in 2015. I will go very fast here because I have 15 seconds. And we screened some active ingredients, and unfortunately, those that gave the best result are neonicotinoids for the mortality of wasp and cage, and also for the time necessary for the first treated hornet to survive and to contaminate the other hornets, because in that case we contaminated only one hornet and then it was passed through grooming and prophylaxis to the other one. So we mimic what happened in the Trojan. One wasp is contaminated and go to the nest and contaminated the other. The third question was how many hornets are necessary for contaminate a certain number of nest hornets. 1 to 10 is a good number. If we treat 1, 10 can die within 1 day. 20 hornets is too much. So, for example, if you have a small nest with 10 hornets, it's sufficient to treat 1 to have the complete neutralization of the nest. So we went on the test on primary nest. We collected the hornets in front of the nest. We contaminated a certain number. We went to a distance. We left the hornets, and they returned and contaminated the whole nest until the complete neutralization. We made 10 and 10 of this test. I will show only 2. In this case, with the medium-sized nest, 1 hornet treated was sufficient to neutralize and to reduce to 0 the nest activity. In other cases, 1 was not enough, because after that, in the following days, pupae survived and new adults emerged. So it was necessary a second treatment to reduce the nest activity to 0. We did also test on a secondary nest. In that case, the number of treated hornets was very high, because in those nests there are thousands of hornets. And we were able to reduce the activity to 0 or to a level that the nest can be removed without problem, because there was a paralysis of the nest. Then we went on the final test. This is the one on the apiaries. We collected the hornets in front of the nest. We went at a certain distance, we contaminated, and then we released them. The first test was done in the framework of this project in 2020 in La Spezia, that at that time was a low-infested area. Now it's not anymore a low-infested area. Together with the region, Liguria, and the Beekeeping Association of Liguria. I will show only one result, probably the main one. We had 3 apiaries and a nest at a certain distance, the 3 apiaries from the nest. At the 3 apiaries, we counted the hornets in front of the of the hives for 1 hour. And then we made the treatment of a certain number of hornets. You see here 25 and 11. In the first treatment, we did not obtain a proper reducing of the pressure, but with the second one, the pressure falls and then increased again, and then with the third treatment, it falls to 0. I will tell you later why this increase after the treatments. But we go to the second and the third apiary. Here the infestation was lower, and one treatment was enough to drop to 0 the hornet pressure. But see what happened at the nest. The nest was in between the 3 apiaries. With the first treatment, the number of hornets entering and leaving the nest in 1 minute went from 120 to almost 0. After some time, it increased again. Why? Because pupae emerged, adults emerged from pupae. Then these were the second and the third treatment at the apiaries, and we in the meantime, we were looking at the nest, and again we see a decrease. And with the third treatment, the activity was down to 0. And this is the nest at the end of the test, so completely empty and inactivated. Then we come to now to the 2023. We are working on a highly infested area, which is the province of Imperia. I will show only 2 of the tests we are doing. In one, we have an apiary and the nest at 80 meters of distance. Again, we counted the wasp, not in an hour because there is a high infestation. This is the glimpse when you pass in front of the apiary and you count, you see 4, you see 10 hornets together in the apiary. Then we made 3 treatments, and you see again a decrease, then an increase, and then a decrease, and with the third treatment, a decrease. What is interesting is that the nest located nearby, it was enough, the first treatment, to decrease its activity close to 0. So what happened there? It happened that probably, this is our hypothesis, other nests in the surrounding area found the apiary and started to predate on that apiary. So that's why the pressure increased again, although at the nest we located, the activity was 0. The second case I show you is 2 apiaries in a very highly infested area. This is the first and the main infested area in Ligury, Bordighera. 2 apiary at 750 meters of distance, and in between a feral colony. We monitored all the 3. You see here, we had to make many treatments to keep low the pressure of velutina, because it is a very infested area. 70 velutine, when you go to the apiary and you count, walking in front of the apiary, you see 70 wasps. And at the beginning, it was over 100. But what is interesting is that with the treatment at the first apiary, at the second apiary, without any treatments, the pressure decrease. So there is an influence of the treatment in one on the other one. But then probably other nests in the surrounding found the apiary, and so it was necessary a treatment also on the second apiary. But on the feral colony, we were monitoring, the pressure decreased and remained low thanks to the treatment at the first apiary. So there is a positive influence of the first treatment. So what is our hypothesis? From literature, we saw also in this day probably that would be 10 nests per square kilometer. So in the area of flight of the velutina, there could be at least 70 nests. This is an hypothesis, a lot of nests. If we work with the method Z in one apiary, we can obtain the neutralization of some nests with positive influence on another apiary. But if we work on two apiaries, then the influence can be even better. And if we apply the method on many apiary in an area, you can obtain the neutralization of all the nests in that areas. So what are our conclusions? That the method is effective and is effective also on other apiaries located nearby, and we saw in Beehero. There is an increasing after some days for pupae emerging by the nest or because other nests found the apiary. In heavily infested area, we have to repeat the treatment more than one time. And if you apply the simultaneous treatment or several nearby apiary, we can lead the elimination of all the nests of the area. But there are also future perspectives, since I also agree that it's not good in a period of reducing pesticides to suggest the use of a pesticide. The method can be applied also with biopesticide or pathogenic agents if the research carried out by some of colleagues here are good results. And also we are studying an automated station, not me, but most and Zan are studying an automated station that recognize automatically velutina, so without manipulation by the operator, and there are applications for other species. Our current authorization is also for Vespa orientalis in Sicily, for example. And of course, we are open to collaboration at the international level if this method is interesting for other colleagues. Thank you.
Thank you very much, Laura, for these exciting results. And we have 3 minutes for questions.
You have a table, you are doing this on the hives.
Not on the hives. Not on the hives, of course, at a certain distance from the hive.
Of the treated hornets in the area, and how many of them arrived against the nest? The proportion of the communities and they come back to the nest?
No, we don't have this number because it's quite difficult to see the hornet with the droplet on the thorax entering the nest. In some cases, when we worked on the primary nest, yes, we were sure that they go in. Not all of them go in, but most of them. Yes, when we work at the apiary, for example, in the last test, we were looking to the nest with the telescope because the nest was far from us, so we could not count it.
Okay, there are some questions from Kennedy. Question 1, when you say 100% selective, do you mean that the method of application applied directly on a hornet is 100% selective, or that the chemical you use is 100% selective? In other words, does not affect any other insect or animals that may eat a treated hornet? This was the first question.
Okay, yes, of course. No, the selection regards the treatment because opposite of a poisoned bait, this was applied directly on velutina, so to no other insects that can. But of course, it can happen that the velutina died and was eaten by another insect. So another animal, yes.
Okay, there is a second question. Would you prefer this method to be used to treat a nest that has been located but may be difficult to reach, or for treatment of nests that have not been located?
Yeah, I think the great advantage, if I understood well the question, is that you can treat nest even if you don't locate it. You can neutralize nest without finding them. That is one difficulty.
Question 3. Hornets likely contribute to a nest from fecal cells even after a nest has been treated. is there not the possibility that this can explain the return of activity at the apiary a week after treatment?
Yes, it can be one explanation, but when the increase is so high, we suppose rather new nests. When there is a very low reinfestation, it can be by new adults also. It can do both.
Okay, there is another question from Tim this time. Imidacloprid resistance is well demonstrated in beetles such as Colorado beetle, and other insects. It is something to worry about in time.
Yes, not now, because now it's not used against hornets, but of course, as I said, if this method would become legal, then there will be a procedure of risk assessment and also the test on this kind of problem, efficacy and the resistance and side effects. Of course, what I want to stress is that we are not doing a test on apiary like was told in these days by some beekeepers. Someone by the Atlantic Positive said 10% of beekeepers are doing this. I think they are much more, at least in Italy, that use this method illegally. We are trying to make this method legal if it will be authorized because it is safe. So I think there is a big difference.
So Tim and Kennedy, I hope you're okay with the answers of Laura. So we can pass over and get directly.
Okay, hi everybody. My name is Zoe and I'm going to present you some results with the electric harps. I'm going to show you how efficient this trap is to protect the hive with limited damage on biodiversity. So to be quick, we all know the negative impact of Vespa velutina on bees for two reasons. First, because hornets are collecting bees. It can be 4 bees per day and even more. But also the presence of the hornet in front of the hive, it's stressing out the bees. And it's preventing them to forage, and then you can lose the colony during the winter because they didn't have any resource. Fighting against Vespa velutina has been quite a mess. Different people try to fight against it from different backgrounds. You had scientists, beekeeper, engineers, local political people or random people that try to find a solution. But actually, so it has been a struggle for many reasons. Nests are too high, hidden, or unreachable, too numerous too. It has been, the fight has been not organized, too complicated, too time consuming and too expensive. So, now we can say that Vespa velutina is an established species, at least in France. And what we should do is to focus on how to protect the hive. So we want a method that is selective. It means that we are only going to kill the hornet, a method that is efficient to be sure that the hive, the colony will survive during winter. We want something easy and cheap. We can't ask the beekeeper to put a lot of energy, time and money in it. And we need something also eco-friendly. So here we are with electric harps. So the first people that sold it, it's people from Spain, from Galicia, yeah, well, from Spain. And today we are using it. It's a local beekeeper that are selling it to us next to Bordeaux. And actually, it's a very simple device. It's a frame, a wooden frame with wires. The whole is connected to a battery, and to have an electric shock, you have to touch 2 wires. It's very important because if you don't touch 2 wires, so there is no electric shock. And we make sure that the distance between the 2 wires is large enough to make the bee able to pass through, but small enough to touch the wings of the hornet, even if the hornet has different size. Why it's also interesting, because if you put this in front of hives, of course you have different insects that are around hives, but Asian hornets have this unique flight. It's hovering in front of the hive, and this electric harp is perfect for this behavior. So we are working with people from Spain, but here I'm going to present you some different results we got with our hives in Bordeaux. So we measure 2 things. First, the selectiveness of this trap by counting and identifying everything that we trapped. So under our harps, we are putting a bucket of water to be sure that we got everything that is passing through the harps. And the second thing is the efficiency of those traps on our hives. And of course, so we measure the weight of our hives. And if the hive is weak, is getting weaker, so it means there is a problem. If a colony is working perfectly, so it means it should gain weight. So the first experimental design was last year. It's very easy. We had 6 hives that were protected with harps and 2 unprotected. So we checked what we trapped every 2 or 3 days, and we followed the weight evolution. And actually, out of everything that we trapped, we got 81% of hornets. So it's quite a lot, and we caught almost 7,500 hornets in 47 days. So that's a lot of hornet. And imagine if we say 1 hornet is collecting 4 bees per day, and if we didn't stop them, I'm not sure we would have still colonies in Bordeaux. But anyway, we caught also a lot of too much bees. But if we check the weight of the hive, we can see that the hive that were not protected, their weight is going down. Contrary to the hive that were protected, the weight is going up. So our first results were quite good. This year we tried to do it a bit differently. We moved a bit the harps just to see if what would happen. So some of the harps, we put them behind our hives. And same thing, we check our traps every 2-3 days, and also follow the evolution of the weight. First thing is, out of everything that we caught, we got 90% of Vespa velutina caught in our trap, and only 7% of bees. So compared to last year, there is a huge difference. We can also explain that because of the weather. Last year in Bordeaux, it was very dry and bees were thirsty. They were just trying to get water and growing in it. Anyway, we caught more than 18,000 hornets in only 36 days. So that's a lot. And what was very interesting, when we compare the harps behind and the harp in front, we realized that the one behind caught much more hornets than the one in front. If we check the weight, same as last year, the hives that were protected, they gained weight, the hives that were not protected lost weight. So as a conclusion, we can say that the harps, are a very good method to support hives. It also, it's not impacting the entomofauna. It's low maintenance. Here on the picture, you can see Denis that is taking care of one of them. So it's very easy to take care of it. It's nowadays beekeepers are using it quite well. It's accepted. It's and it's also like a way to not harm the environment. Also, it's very important nowadays. And what we would like to do is to find a solution to kill or collect the hornet, but without using the wet bucket, the water bucket. So either using a stronger electric shock to be sure that the hornet is going to die on the spot, or wet collector, a dry collector, sorry. What we would like to do also is to customize the harp, change the colors, the position and all that to see how it impacts the trapping. We also observe the behavior of the hornet. It was very interesting because you have some of them that are in the front of the hives and they are hovering, and some of them are at the back. So we need more study and we need to understand what's going on, like what is this hunting behavior. And finally, we have also to think about the weather, because as I said, last year was like a dry season and the hornet came out, well, we trapped them later. This year was a bit more wet, and in August, it was a crazy pressure on the hives. So you can see here, the difference between the 2 years. Thank you very much for your attention. Thank you all the people that helped me for that.
So thank you very much. Perfectly in time with compliments of the chairman for this. And there are 5 minutes for question.
Question is out, right? But I have a question about the actual cost. I mean, the maintenance cost, especially the hard cost, like 120 euros, something like that, right?
Mm-hmm.
But then you put it in the apiary, and then it's just that. I mean, you have to. I don't know, there is a solar panel.
So, like the first year, we didn't have a solar panel. The second year, we used one, and it's very efficient. It's very easy. Like
Okay.
you can, if you like how to build things, you can make it yourself. Also, you have everything on the internet, so it's like very easy. We choose to buy it from a local beekeeper because we also know them, and I think, well, it was a way also to support them. But it's very easy to handle.
Apart from that, there's no other maintenance on the farm.
Well, like the thing is with the water collector, you need to change it because at some point, if you have a lot of pressure of hornet, it's like filled with hornet. So you need to change the water.
Okay. And just the last question.
Yes.
Have you measured the number of hornets? I mean, the reduction of hornets, apart from the one that you are able to catch in your traps. How many do you have in front of the hives? Because you know, of course, like colonies produce every day,
Yeah.
workers. So interesting to see if there's an actual reduction. But of course,
So yeah.
you keep a paid population, but still, I mean, Can you see a visible
So-
effect of the injuries?
When you are next to the thing is, we put the harps because like the pressure was high, and it's very impressive because as soon as you put it, you see the pressure
Decrease. Yeah.
decreasing, sorry. And you see also the bees, the ones that were not protected, you had this massive bee growth. And the one that were protected, you didn't have that. They were like coming, getting out and coming back like usual. So it's like, you can see it as soon as you put the harps.
Brilliant. Thank you very much.
Hello, thank you. How many times the electric arc stopped working? Sorry, if you have any problems with the electric arcs in terms of working in the appearance.
We had some. So actually, where we have like other hives a bit further, and we put our harps, other harps next to those hives. And we had some trouble because we were not going there that many times. So at one point we noticed that one of the wire was broken. So it's also like you have also to check sometimes, of course. But I've heard like a lot of people had trouble on touching wood. For us, it has been working quite well. Even if like Bordeaux is this year, it has been very wet. Wet weather, we didn't have any problems.
And in the season, from where to where we are putting the arcs, the electric arcs.
So like that's the thing. Like it depends. Like I mean, it depends where you are. If you have a hornet nest around, it's like people. Beekeeper has to like check, observe, and maybe like adapt how to do it. Last year, this year, and like we are working with people from Spain, nothing was the same. So that's why like you have to be aware of what's going on around and adapt yourself.
So we have two more questions. One is from Annabella, the other one is from Kennedy. Annabella said, our experience in Portugal is that with water receptacles in areas of very low relative humidity and little water in the region, many bees are trapped. Have you tried it compared to dry receptacles?
So like what we tried is to put some like different things in the water to make sure that the bee would not go inside, would not be attracted, and well, nothing was very efficient. So we didn't find anything for that yet. And I know people are trying to develop a dry trap under to be more, that would be more efficient. I think Sandra talked about it yesterday. So we have to find a solution for sure, but now I don't have any suggestion.
Okay, thank you. Then Kennedy, is there any indication that with time hornets will learn to avoid electric harps?
That's why we changed it position. We changed the position, and no, actually, like we think if the hornet is passing, is like touching the harp, it's deaf. He's not going to learn. He's dead. So I hope not. But I mean, like changing position is also like a good way to make sure that they will never learn or never see it.
Okay, thank you very much. So we can pass over. And there is Gemma Burbui. So Gemma Burbui from University of Pisa. And she talked about the fight against Vespa velutina in Europe, an assessment of surveillance, management, and knowledge transfer across borders.
Thank you very much. Hello, everybody. And I'm going to talk about, obviously, Vespa velutina. And we all know well the issues related to that species. After 20 years, the first report in Europe. Despite all the studies about the species, we believe that there are still some issues related to the Vespa velutina management, despite being a listed species on European Union invasive alien species regulation. We believe that there are still some issues that necessarily must be resolved to allow us to avoid the major spread in Europe of the species and to avoid and tackle the species. First, there isn't yet a clear and complete overview in Europe about Vespa velutina situation, both in terms of species spread and population dynamic, and also in terms of management objectives and techniques applied at the European level. Second, there is little collaboration on surveillance and management across European borders. A good collaboration could allow us to prevent and avoid the arrival in new countries of species of Vespa velutina. Finally, there isn't a formal mechanism for knowledge exchange and data sharing that could allow us to have a pan-European approach against yellow leg hornet. So thanks to an initiative of the Belgian Research Institute for Nature and Forest, INBO, we created an online survey to study the European situations of Vespa velutina, and also to, we hope that, try to resolve these 3 points. A very similar study has already been done, but on a smaller scale, just few regions in Portugal, Spain, and France, and with a different target audience, just includes beekeepers. Our aim is to work on a bigger scale, not just national levels, but we want to work on a regional level, considering all the regions in each European country. And we also want to work with a different target audience, including all those people involved in Vespa velutina's management. And our survey is still ongoing, so the aim today is to try to have some feedbacks from COLOSS task force and also from you, all the people present at this meeting, to improve that survey before sending it to our target audience. Our main aim is to highlight the diversity of approaches in terms of velutina management approaches and techniques applied at the European level across borders, and also to have an overview of stakeholders involved in the whole process. We will also want to gather some data and indications about knowledge gaps on management, and also some indications about all the costs incurred in the whole process. At the end, we want also investigate perceptions, people perceptions, particular experts about Vespa velutina, about perceptions about the species itself. So, our target audience, as I was saying before, include all those people involved in the management of the species, such as people involved in the implementation of invasive alien species regulation, project managers and officers in the domain of environment, bee health, public health, and agriculture. This kind of target audience will allow us to get and gather very precise answers, considering that some questions are pretty precise and specific. We don't want only to work at a national level, but also at regional and sub-regional level, considering, like I was saying before, all the regions in every country, European country. The survey includes 5 different sections, and it will take more or less between 15 and 20 minutes to be filled out. The first section includes questions about the respondents' professional data, so obviously not personal data, just professional. And there will be also questions about invasion status of Vespa velutina in respondents' country and regions, like when the first report was, or the introduction pathways. The second section includes questions about respondents' perceptions about 3 different factors about Vespa velutina. The first, respondents' perception about Vespa velutina as listed species in the regulation. Second, respondents' perception about Vespa velutina impacts, like the different weight and importance that different respondents could give to different impacts on biodiversity. And finally, perception about Vespa velutina management. This kind of questions is linked to the 5th section that is about management, you will see. And these kind of questions allow us to link and compare the management, the current management in each country, to respondents' perception about it. So if it's work or not. The third section is about surveillance and monitoring systems. And we are interested in, for example, how they are organized with spatial level and who is involved in this process. 2 minutes. 2 minutes. The fourth section is about nest detection. And also here we are interested in people involved, the techniques applied, and the proportion of nests detected. Obviously, it is a guess because we are not able to have the exact number of nests on the territory. The last and 5th section is about management. And here we are interested in objective management, objectives, people involved, techniques applied, and the budgets of the management, the knowledge gaps we could have in the process, and the percentage of removed nests. So this is the structure of the survey, and my main aim today is just to ask you your feedbacks about the survey, because we would like to improve it before sending it to all the countries. So in the next slide, I'm going to leave you the link and the QR code for the survey, and I'm going to ask you to fill it out. It will take more or less 15 minutes. So in these days, maybe you could be able to do that. And at the end of each section, there is a question and free space where you can put your comments, ideas, suggestions, everything that blew up to your mind, like if the questions are clear or if something is lacking or whatever you think about the questions and the survey. And more important, very important things, I'm very interested in having contacts of people at European level that would be interesting to fill the survey out. We will need so much in having contacts. So we can have at least one contact person for each region in each country. So this is the link, the QR code, and there is also my email, my contact. So you can write me an email if you are interested into having the survey when it is ready at the end. And you also come to me in these days and talk to me and ask me everything you want about the survey. Thanks.
First of all, I think it's a great idea to start with, especially with things like costs and management experiences. Just an idea, I'm not quite sure if topics which we in this group cannot answer, especially biodiversity, should be topics asked in such a survey, because it seems there is no answer yet, and the perception is, yes, a very unsubstantial answer. And as the survey said, 15 to 20 minutes, it's really long. So my suggestion is concentrate on the questions that people really can answer and make it a little bit shorter. It's just a suggestion.
Okay. Consider that the perception is just personal thoughts of the respondents. So just to have an idea about what expert people think about Vespa velutina situation and impacts, more or less.
Our perception, not only the fact that, as Ina was saying, is subjective, but also depends on the context. I mean, in different regions, in different countries, you have different velutina pressure, you have different pressures. So, I mean, have you thought about how to control for the different situations and how they can affect the perception of the people?
Yes, of course. I think that based on the kind of respondent, there could be a different answer, and maybe different perception will allow us maybe to understand the objectives of each country, maybe. And so different countries with a different level of the problem has different objectives, and maybe these differ in perception.
Just at one point, each answer will be linked to a specific region, so we will know this perception comes from Tuscany, this perception comes from, so we know the link between the situation and the...
Without microphone, the rest of the people,
I mean, we have, for example, evidence of the actual impact. I mean, there are many things that we don't know. The number of nests.
So we divide, I don't know. And so we will know that.
Yes. Each question has different answers. So if you don't know how many nests do you have on your region, you can put, I don't know. If a country like Austria doesn't have the hornet, they can put, we don't have the hornet, so they can answer.
the external output will not.
The question was if the people ask the question, we know about the actual situation in that particular region. That's the question.
Yeah. I think it's why we are going in first section to ask the invasion status of the species. So we can understand all the answers that the respondent puts in the following section and questions.
I don't know if I get you correctly. Are you asking about a perception or real data? When you ask about cost or proportion of nest, you ask the perception of the fish. They get real data.
The perception is just in second sections. It's separate section. In all other section, we are going to ask about data. So the precise number of nests, or more or less a guess, because sometimes maybe people don't have the precise number or the precise data about the question.
Then a suggestion is that use the plan start to compile the survey, because I think that after 5 minutes people became there, so taking that my personal.
Thanks.
I agree, but remember that people who answering should be the ones involved in management species. If they cannot spend 50 minutes to fill the survey, I don't know.
At the end,
Let me feel them.
at the end, we hope to be able to give to each country some suggestions to improve management approaches and techniques. So people should be interested to have the data back when we analyze the responses of the survey. So we are going to work with expert people so they are able to answer, I think, and should be interested.
So what I think is actually that you actually get like 2 different questionnaire surveys out of that, keeping in mind who is the person who's going to answer. In the sense that if you are talking to people which are experts, then you can get the official data. But in my opinion, perspective of normal people, it's also so important, because like with our small projects on Vespid plants, normal people actually are more sometimes interested to participating, and they become a really great resource for scientific projects because they are actually excited about being part of the scientific community. So what I would suggest you is also to make it shorter. Try to get, so like if somebody is scared of a hornet, then definitely that's an important fact for you. Also to build up a study and create that kind of discrimination that I believe the scientific community needs to do on the territory. So think about who is your target person in the end, and try to reduce the number of questions and have a focus on who is your target at the end. But try to involve the normal non-scientific community because they are a great resource.
Okay, I'm going to answer you to that comment, just saying that normal people ideas are very important. I agree with you. But maybe in that case, we will need a second survey, a different survey, because if we just want to have an overview of European situation, a clear clear overview of European situation, we need very specific data and answers. And maybe just expert people could give them to us, I think. But I agree with you, obviously.
So good evening, everyone. To change a little bit the theme that we're talking, I'm going to talk about a different Vespa, the European hornet, Vespa claro, and also Vespa orientalis. So as you may know better than I do, Vespa claro is a eusocial Vespid native to Eurasia, and whose range spans from Japan to the Iberian Peninsula, and is a semi-generalized species that feeds in many types of arthropods, including but not limited to the honeybee. But why study it in Sardinia? It's because Vespa claro was the first hornet to be seen in the island in 2010. It was, and also Sardinia being an island, it's a very much more delicate ecosystem than the inland. We were able to carry out this project thanks to the ALM and ALM apostrophe interregional projects. Our main objectives were to define the distribution of Vespa claro in Sardinia using verified citizen science and testing its accuracy, creating a monitoring network for Vespa claro and also a surveillance network for Vespa orientalis and Vespa velutina in Sardinia. Also, we tried to assess if Vespa claro showed any invasive traits in its new habitat in Sardinia through behavioral observation of nests and beehives. And finally, creating an expansion model of Vespa according to climatic and environmental whatever. Okay, so with the help of citizen science, we were able to retrieve 125 reports, 101 or 80% of them being correct of the presence of Vespa in the island, mainly in the northeast part of the island, but never from the south, thus verifying its absence in the south. With that information, we hypothesized that it may have come from yam cargo arriving to the port of Olbia in 2010. And of that 80%, 95.1% of the reports on hives or nests were correct. And as for the individuals, 73.8% were correct, which is considered viable for ecological values. But we have to keep in mind that Vespa was the first hornet present in the island, so the only species that could cause the citizens confusion were Megascolia or Scolia and Eumenes. With that, we were able to verify that citizen science is available too for monitoring alien species, but as well as for surveillance, because in 2021 we first reported the presence of Vespa orientalis in the south of the island in Cagliari. And as a follow-up research, we carried out a behavioral observation study on Vespa crabro, recording 6 nests that were provided by citizen scientists again. And what we tried to assess was if the Vespa crabro showed, like I said, any invasive species trace in the island. From the foraging activity photos and videos that the citizens sent us, 50% of the time the wasps were foraging fruit, followed by sugar exudates, then flowers, bee predation, and lastly water. But from the recordings that we had on the hives, 70% of the time, we recorded 17,500, roughly 500 forager wasp flights returning and going out from the hive, and 70% of the time they came back empty-handed to the nests, followed by 15% of the time where they retrieve unidentifiable pieces of food, pellets, followed by material for nest building and repairing, and lastly, with 1%, they retrieve identifiable prey. Of that identifiable prey, 41% of the time were grasshoppers, followed by mantis and dragonflies. So you can see Menoptera are quite low in the scale, with only 10% of them. But since 75% of the time they return, 15% of the time they return with pellets with unidentified food, we also wanted to test its behavior near hives. With that, we retrieved from 3 recordings. 2 of them, I forgot to mention that of the 6 recordings, 4 were of nests containing the brood, the queen, and the workers, and 2 of them just the queen and the brood. So for this study in hives, we used 3 recordings for 2 of the 2 breed and brood, queen and brood nests, and then 1 of the worker queen and brood nests. So as for the 2 queen and brood, no attacks were recorded, and for the other one, 21 attacks in the span of 8 hours were recorded, 19 of them being against forager bees when they were returning to the hive, and just 2 of them direct attacks on bees. One of the time they responded by volley, as you can see in the image, where they suffocated the wasp, the hornets, and they dispose the body. As for now, we're still working with citizen scientists on Vespa orientalis. In fact, we've reached 450 correct reports of Vespa crabro, always in the northeast of the island. And also, we are, like I said, we first recorded the presence of Vespa orientalis, and we have now reached 150 reports, correct reports of Vespa orientalis only in the south of the island. And we started working, doing the same work that we did on Vespa crabro on Vespa orientalis now. As for the modeling, my colleague who is in charge created this heat map, where you can see the probability of the presence of, like the suitability, sorry, of the different areas of the island for the presence of Vespa crabro. And as you can see, it follows the current distribution. In North Sardinia, it is consistent with our reports from citizen scientists, but other highly suitable areas are almost all of the coastal areas and a few other restricted areas in the interior. So I've been working with this group for like 6 months. I just started my PhD, so I will be happy to answer to as many questions as I can. But otherwise, for further details on data collection, you can ask, leave an email to my tutor, or for further details on the model, you can contact my colleague. You can also, of course, check our published papers, and that's all. Thank you for your time.
It is a comment, so you can think about this. In the slide, when you showed the foraging behavior, could you please come back?
See. This one? Or
yes.
This one?
Yes. Maybe this nothing can be meaningful because maybe they are not bringing anything on the mandible or the legs, but they are bringing water, nectar, honey inside. In fact, we made some experiments, and Peter said some about that. They can gain until 100 milligrams to eat on a carbohydrate source. 100, that is almost the entire weight of the hornet gaining and coming back to the nest, which we don't see any.
No, in fact, as you can see, like 50%, half of the time they were feeding on fruit, so and also sugar, so most likely they were bringing some carbohydrates inside, yeah.
Slide where you mentioned that very few, yeah, exactly, there's very few piece where we catch. But what I find very interesting is, for example, the high percentage of mantis. And mantis often is an endangered species. So here you could have the connection between an alien species and predation on endangered species. Maybe also with the very glass eventually. So did you check on that?
Yeah, we'll check. I think for this data, we had like 39 videos or photos. So it's not really a big number. And it's also 1% of the things that they retrieve to the hive. But yeah, you can surely look into that.
Okay. Keep going. Can you hear me?
Okay.
European hornet is on Corsica. So to what extent is it really alien to Sardinia?
I don't know. I mean, it hasn't been recorded ever, so I mean, the data that we collected suggests that it is not invasive, it's alien, because it shows the same traces in the native habitat. So we don't consider it invasive, we just consider it alien.
Sì, bene. Thanks. So just thanks, especially to Anna and Karen. We just would like to switch the order of the presentations. Well, so climate changes and globalization. This is a presentation that I did at the Congress in Hamburg on allergologists. So just I have to change some, but just it's important to introduce also the next speaker. Well, so climate change and above all globalization have a big effects on the life of many animals and plants and living things. Okay, and especially on insects, he has a very strong influence. So there's a reduction in biodiversity, changes in the distribution ranges, and competition of alien species with original species. And here we are mainly interested in distribution changes of those insects that represent a possible health risk for humans. So, social insects, we have to speak about that because they are scarce in number of species, as you see here, but their percentage in biomass is above one half of more than one half of all the insects just described. Hymenoptera in this part are a large group, and they have at most 2,000 species. And in the Aculeata, which is a section of the Hymenoptera, we found a number of species which have glands producing venom and a sting to deliver it for chasing prey or just for defending their colonies. In some cases, these social insects present colonies with millions of individuals. Social Hymenoptera, we find social species in the ants, as you know, in bees, in family Apidae, and in wasps, family Vespid. All these insects defend their colonies with venom and sting. And they present colonies in various size, and which span from a limited number of adults to millions of members. Aculeate Hymenoptera, in particular the social one, can cause serious toxic and allergic reaction to humans. So new species have introduced in new lands and have enlarged their distribution range due to commercial trades and to globalization, of course. And climate changes just can cause just the enlargement of the range of species in various areas, increasing their areas of presence. All these things, I mean, just globalization and climate changes are due to human actions. And the provisional models of the dynamics of a distribution range in various invasive species are necessary to predict the manifestation of an alien species in a given territory. And so it is important to be prepared to face new sanitary challenges from this sort of the new species just will front in the future. Just speaking about invasive stinging species, we have, for example, Formicidae, which are very, very good invasive species because they are just very can just acquaint to a new situation, just a new climate, and so on. The most known species of ants, alien species of ants, are Solenopsis invicta, for example, the red fire ant, which just is from South American origin and invaded many parts of the world. Solenopsis geminata is another one, just concerning the east part of the world. And in the more just known examples, we can find also the Brachyponera chinensis, which is called the Asian needle ant, which is also very invasive, especially in the eastern part of the globe. Speaking about Solenopsis invicta, you see that this in the Odiern distribution area, and you see that from South America, it just colonized many other parts of the world, but it's the potential global range expansion of the invasive fire ants. And as you see, just all the world is in fact comprised. If we just go to the quite recently discovery, we found that the very first bridgehead in Europe has been also already established in the southern Sicily, just starting from here. There's a sort of airport where probably just the very first queens just started. And also this is a very challenges that we have faced in the next years. From all the apidae, we can mention the Africanized honeybees coming from a sort of an experiment just tried in Brazil, just trying to find a sort of well, just invasion of this Africanized honeybees, just caused by man also for this experiment, and they just invaded all the south of United States and Central America. But Vespidae are also the focus of the invasive, for their invasive capacity, as the foundation of the nest is usually performed by a single queen, and that can be easily transported all over the world by commercial trades, for example. Here we have in Vespidae, we have two main subfamily, which are social, the Polystine and the Vespine. Polystes dominula is belonging to the Polystine, and also Polystes olivaceus. And this was just represent a quite common paper with almost 2,000 species in the genus. And they are present all over the world and form annual colonies in temperate countries with a maximum of a few hundreds of rodents. Polystes dominulus is a very invasive species. He had colonized starting from Europe, the United States, and in the south of America, Argentina, and and in Chile, and then in South Africa, and in Australia and New Zealand. While Olivaceus just is colonizing a lot of countries in the Southeast Asia and around the Indian Ocean. Okay, 1 minute. Okay. West, between wasps, we can mention Vespula germanica, but in the Vespine and the invasive hornets, there are the most important and documented reports in the entomological literature. Crabro, orientalis, velutina, but also Vespa tropica, Vespa bicolor, and Vespa mandarinia. This is just passages from the main, just original area in the Southeast Asia, and just arrived to try to influence many countries. If you just give a look to the cladogram, the phylogenetic point of view, just a cladogram of the Vespide and Vespine, we see that mandarinia and tropica are quite near, Crabro and orientalis also, and bicolor is near Vespa velutina. The case of Vespa velutina nigritoris have been already just spoken about, and in Tuscany we have a special situation, as you know, that because Vespa velutina is coupled by the invasion of Vespa orientalis coming from the south. And so we have in Tuscany, we have 3 species just sometimes living together. But this also brings new sanitary emergency, also presented by Vespa velutina nigritoris, for example. Toxin and allergens contained in the venom can cause organ dysfunction and fatal allergic reaction. And we have many casualties in various dozens of them in Europe, Spain, France, and so on. And principal allergens are this Vespa Vu5, which correspond to allergen 5, and Vespa Vu1, which corresponded to A1 phospholipase. Plus, there is some hyaluronidase isophones. High homology with the Vespa crabro and the Vespula species, lower with Polistes dominum. This is speak about the venom and the possibility to treat the allergic person with some vaccines just made with the venom of this sort of wasps. But of this, we'll speak about Elisa, which follow me in the next speech. So you see again this sort of cladogram, and this is just to remind you that the near just Vespa crabro and Vespa orientalis are quite near, and also the venom is quite near. So mandarinia and tropica, which are oriental mainly, and bicolor, and the velutina is just something like different because it seems to be just very far from other, with the exception of the Vespa bicolor. Okay, conclusion. Various species of stinging ants, bees, and wasps with allergenic potential have invaded new territories, and predictive models foresee an increasing this tendency in the nearest future. Specific recognition patterns of the sensitization for the venom must be developed to counteract allergic reactions, two stings of the species of these new species with appropriate therapies. And of course, the recent invasion in Western Europe of Vespa velutina and Vespa orientalis are example which open important problems for the treatment of severe allergies. Thank you for attention.
So I'm the outsider here. I'm a doctor. I work in Florence in an allergology department. And this department has been dealing with this topic with Hymenoptera venom allergy for the latest 30 years.
Go.
Okay.
Sorry.
My speech will involve an overview on Hymenoptera venom allergy, both apids and vespids. Because they are all a threat for our health, okay? So, okay. Being stung by an Hymenoptera is a common experience for a human being, and up to 30% of the people stung will produce antibody, IgE, also other type of antibody, but I speak about the IgE because the IgE is the allergy antibody. This is a mistake made by ourselves, but it's not enough to label a patient as allergic. So having IgE is a condition to make a diagnosis, but is not enough. There has to be a reaction. So 1/3 of the person in the world are sensitized, so they have IgE for venom, Hymenoptera venom, but they will not have reaction with further stings. So they have to do nothing. Only a few persons among these 30% will develop a systemic reaction or any kind of reaction, allergy reaction, and a little part, fewer than this, will have life-threatening disease. So a systemic disease that poses their health in danger. Fortunately, death among allergic persons are rare. This could be the first sign of allergy. This is called large local reaction, is a swollen red warmth and itching edema area that is around the stung site. Okay, why I told that this could be an allergy, because the test, blood or skin test, has to be positive. In the case of negative result, this reaction has to be considered as irritative. Okay, so this is but this reaction has to take the patient, the person, to a medical advice, to seek for a medical consultation. This reaction can happen immediately in the first hour, or can be delayed. This is the same. Okay, it could be allergic in its nature otherwise. Usually it can be treated with corticosteroids, antihistaminics if the itching is high, and it takes a few days to resolve. They don't need any antibiotic. And the first, this is a study where I participate, large local reaction. After only once large local reaction, the risk of progression is a little bit more. The study indicates 25%, but with several large local reaction, the risk, the rate of progression to other dangerous clinical features lower down. Okay, clinical presentation. This type of allergy can present with several, of the whole spectrum of features, from the large local reaction to skin involvement, to gastrointestinal involvement, respiratory and cardiocirculatory, with syncope and collapse, like all other allergies. The whole thing starts with an IG that recognizes the venom and that activates mast cells. There are immunity cells that are all in our skin and other organs, and when they are activated, they can cause allergy and anaphylaxis. Who has to come to our division, to an allergy division, and when? Who, we said, at least he has to have a large local reaction. No familiarity has to bring the patient, a story of a father allergic has to bring the patient to our observation, because the tests are has to be done only when a reaction has been present. When? As soon as possible, then we will manage the time to do the test. So an accurate history is mandatory to recognize the culprit, so the insect that can have provoked the problem. We perform a skin test and blood test. The skin tests are more sensitive and reliable. We give a patient an action plan. We explain to the patient what he has to do when he is stung. So the first hour is the most important, because only in the first hours severe reaction can happen. Avoid physical exercise in that hour. Be ready and know how to manage the therapy, the emergency therapy. Choose the proper therapy based on the clinical symptoms. So corticosteroids and antihistaminics in skin involvement, and epinephrine in the most severe happenings. Patients are suggested, patients who had severe reaction are suggested to bring with them epinephrine autoinjector. That are available in Italy, 3 types at the moment. But the therapy, the emergency therapy is not enough, because it can be used only when the reaction is present at the moment, okay? While immunotherapy prevents. Immunotherapy is subcutaneously administered therapy that is suggested in the most harmful reaction. It's established, is effective with a high degree of effectiveness, and safe in the proper setting. That has to be an hospital setting by trained personnel. It has a long administration period, at least 5 years, to maintain the benefits achieved also for longer periods. Okay. What is interesting for us is to choose the proper immunotherapy, and so for us, venom can be divided in 4 classes, major classes, that are based on the cross-reactivity, so the similarity on the venom. So Vespula and Vespa crabro are quite similar. So often we can vaccine the patient with one of the two. In Italy, we have Vespa crabro immunotherapy, so we don't need to vaccine with Vespula, but in other countries they perform this kind of operation. Apis going by his way, and also Polistes. The problem is when we have the sensitization to Vespula and Polistes, and we don't know which is the culprit insect. So we have other arrows on our arch, but I will not talk about this. Only a few things about this. When Vespa velutina entered our country, we were worried about the efficacy of Vespa crabro venom or Vespula venom immunotherapy, because we didn't know if this would cover allergic patients to velutina. But with the CAP inhibition studies, there is a blood test, time-consuming and onerous, but that can help determine the right venom to give to the patient. And we see that Vespa crabro venom immunotherapy is enough to cover that patient allergic to velutina. We have at the moment 2 patients vaccinated for Vespa crabro that were stung by velutina. And so at the moment we are satisfied. Maybe in the future we'll have, maybe, I don't know, a vaccine specified for velutina. And only one thing, with the multiple stings of velutina, we know that we can have an important, also important toxical effect, more severe than the other Vespids, because they have hemolytic substances, myolytic substances, and some other compounds that are active with on the coagulation, so they can have organ failure, important organ failure. And stop. Thank you.
So now is the turn of Anna Granato from the Zooprofilattico Institute of Padua. And she will shift to another species, Vespa orientalis.
Good evening to everybody. The afternoon is already past. I would like to present the oriental hornet as a potential vector of honeybee pathogens and a threat for public health in Northeast Italy. As you know, because in this day we work a lot about that, global warming and the globalized movement of people and goods are the main factor that are facilitating the expansion of historical distribution and the colonization of the new territories by Vespa orientalis. Vespa orientalis is native to the Central Asia, India, the Middle East, northeastern Africa, and the southeastern Mediterranean area. Vespa orientalis has recently colonized several European countries. It recorded in 2012 in Spain, 2019 in Romania, 2021 in France and Greek Island. Vespa orientalis is also introduced by human in Chile, Mexico, and China. In Italy, Vespa orientalis is well established in Sicily, Calabria, Campania, and Lazio region, and recently recorded also in Sardinia, Liguria, Friuli, Venezia Giulia, and Tuscany. If you think that Vespa orientalis adult feed on carbohydrates and sugary substances, and the oriental hornet larvae were fed animal protein, mainly represented by large-bodied insect, in particular honeybee, but also by fresh or spoiled meat and fish. You can understand that the hive represent the preferred food source because contain both protein of animal origin and sugary substances. The great impact of predation on honeybee hives occurs usually from July to November, coincide with the exponential growth in the protein demand by larvae. The negative impact of Vespa orientalis on bee health and consequently on bee production is not limited to the damage caused by direct predation of bee, which leads to weakening of the colony and sometimes to its collapse, but also by the capacity of these species to act a potential vector and spreader of pathogen of honeybee. For this reason, we would like to monitor the presence of pathogens carried by Vespa orientalis that could potentially threaten honeybee and public health. A very simple field experimental was carried out in the province of Trieste, not East Italy, because this city has only recently been colonized by these hornets. That's arrived probably by goods, by goods unloaded from ship by in the local commercial port. 3 experimental apiaries were set up, each consisting of 3 Dandenblatt hives with 3 bee colonies, and they were located in different position and altitude. Apiary 1, identified as urban, was located in a park in the center of the city, in the center of Trieste. Apiary 2, identified as Suburban Hill, was located in an inhabited but a peripheral area of the city at about 200 meters above sea level. And Apiary 3, identified as Cass Wood, was located in the thick of the wood in an uninhabited area 4 kilometers away far from the city center and about 300 meters above sea level. In these slides, you can see the trap that maybe you can this with this no neanche. See, you can see. Okay, this is the trap that you used in the neighbor of the apiarist. Che mi sono intrippata. Ok, scusa. Grazie. This is the trap that we use in the neighbor of the apiaries, and we use to attract a hornet salmon cat food. The catches was performing in the spring in order to capture the founder's queen, and during the summer in July to capture the workers. All the horns were subject to gross examination to exclude the presence of lesions, and then froze immediately before sent to the laboratory. At the laboratory, the samples were macroscopically examined to evaluate the possible presence of morphological alteration or parasites like Varroa. And in this table, is you can see the pathogen investigated and the laboratory technique used for detect them. You check the presence of Nosema spore, a fungi, 7 viruses, 2 trypanosmatids, Lotmaria passim and Critidia mellificae, and aerobic bacterium. While for parasitological and viral examination, its samples was analyzed individually. For bacterial isolation, the analysis was performed on a pool of individuals. Bacterial species identification was performed by Malditoff, and the result of this analysis is expressed with the full name of the microorganism being tested, accompanied by the identification reliability score provided by the instrument. First, the catch were performed only in apiary 1 urban and apiary 2, because Vespa orientalis has never been detected or seen or detected and captured at apiary 3, Caswood, at 300 meters above sea level. I will add to remind you. 10 queens and 5 workers were captured, and of the 10 queens, only 4 were considered suitable for analysis. Non-morphological alteration or parasites were detected, and all samples, queen and workers, tested negative for Nosema spp spore and for Lotmaria passim and Critidia mellificae. Sì. Concerning the viruses, as you can see, 5 of 7 virus investigated is present in workers in orange, and only 3 in queens in blue. The most detected viruses is BQCV, present both in queens and workers, followed by SBV, present in all workers and in 3 out of 4 queens. The former wing virus is present in queen and workers, but not in all samples. And ABPV and CBPV were detected in 1 and 2 workers respectively. IAPV and KBV was never detected. The rate of co-infection that is shown in this table is very high in this sample of Vespa orientalis, and this situation is sometimes more than sometimes frequently in honeybee in the hive. And the honeybee virus identified was the more frequent virus detected in apiaries across the territories, suggesting that the passage from honeybee to Vespa orientalis occurs by predation. A predation of honeybee adults and larvae infected, and the cannibalization of the carcasses. Our results confirm the recent results that are already published by Karen, that is the speaker after me. About bacteriological examination, bacteriological analysis, this table show the results. The reliability of the identification analysis performed by Malditov must be interpreted with the legend present below. No Salmonella and Listeria were detected in both in queen and in workers. Enterococcus fecalis is detected both in queen and workers, and
between what was told about mandarinia and what Vespa does. So it goes in front of the hives, catches and then flies away. But as soon as you open it, like you open a hive, they're not interested so much anymore in the bees, but they're super interested in the larvae. So one of their, and so also like one of the ways that actually works quite well is to put brood. So what, like some of the video methods, they put like brood inside an empty hive, so it works like an attractant, and they're not interested anymore so much into the actual colonies where bees are. And then there's a second question. Nested sites, primary and let's say secondary nested sites, they are in cities. They love cities because they like dark, not humid, but they like dark environments. So you will always find them if it is little holes in the wall, in buildings. But one of the, like where you have air conditioning systems or where you have boilers, so literally you have them outside the houses. So that's where you will have them, or you will have them mainly. So where you have the, so you know, no, I can't remember the word in English. It's not the window, you know, when you get the shutters. So it's a shutter box.
Also,
and that makes them the ability also to come inside houses.
And also under the ground, or no, against the body trees on the continents that you know, but the nest also on the interface the colonization nest, those usually nests are on that in the church of the day that is saying okay, it's only 900%. So maybe I mean it's not, it doesn't look like
okay, we have also a question. There is no time. As you can see, there's nobody asking question. The question is for Anna. As a top predator, could the pathogens determine not just reflect bioaccumulation by preying on a wide range of prey, including honeybees, wasps, and net pollinator, which are already transferring pathogens amongst each other? And then, were you able to determine whether viruses are replicated in Vespa velutina?
Yes, this is
Vespa orientalis, not Vespa velutina. Okay.
Vespa orientalis. I say that is a potential vector. I don't have evidence that there is a transmission between Vespa orientalis to honeybee. As you can say, that in these samples we found the honeybee viruses. Okay, but I can see that it's a potential vector. I answer, no? Okay. I don't have evidence, and maybe some people, I don't know if some people study about this, but I don't have any information about this. I don't know if you have maybe Karin about the replication.
Yes,
in
actually now.
You are studying now. Okay, so the study is ongoing. Yes.
The last one, because
Yeah, so besides the vector, it's also interesting is the three common viruses that you found, the paralysis and the black queen and the cold queen. Are they symptomatic or do you just detect the?
Just detect, no symptomatic, no alteration, no deformed wing, anything, just detect the presence, but no morphological alteration and no parasites. We don't detect, for instance, varroa.
So Karen Power from the University of Naples about the Vespa orientalis emergency in Campania.
So first of all, I'm going to tell you where Campania is. Probably not everybody knows. It's in the south of Italy, and it is one of the regions in which Vespa orientalis is actually super well established. It's so well established that it really likes being there and feeding on hives and bees and killing nearly almost 50% of our colonies. So what you can see here is just a normal day in an apiaries in the Campania region, loads and loads of Vespa orientalis doing what they usually do, so predating and going inside the hives to steal and rob everything that they can actually steal. So considering the emergency that we are looking at in the past couple of years, I would say, we finally managed to have funding. It's not a big fund because I'm talking only about 15,000 euros. And with this money, which came from the Campania region, we actually think we did quite well with 4 different activities. So starting from monitoring, trying to understand how many there were and what were the relationships that were actually happening between Vespa orientalis and honeybees, Vespa orientalis and other kind of animals, Vespa orientalis and the environment. So this obviously responds to the need of having actual studies and research of how this animal is actually behaving in our new, in his, let's say, new conditions. I say new because obviously something has happened. It has been well established for years, and it is considered as native, but there has been something which actually switched it to becoming a problem. And this is something that we don't know and that actually we need to study. Then information that we take from here and from obviously the projects, which are research projects ongoing, are the basics of actually finding resources to control and to fight this problem. Last but not least, dissemination. So not only, as I said before, with the community, with the scientific community, but also with what I say, the like population. So considering monitoring, in order to save budget, we worked with a previous network that we already had that we used for checking the health status of colonies. So we are talking about 5 colonies for each of the provinces that we have in the Campania regions for a total of 25 different apiaries. We had connections and we talked with associations in order to have data from them. So actually what was happening in different apiaries. And then we used Citizen and Science to see what the population was actually thinking about and if they could actually help us. Considering the monitoring results, so actually considering what was happening in our apiaries, in the province of Naples and Caserta, we had a strong presence of Vespa orientalis, as well also in Salerno, but we had the absence of Vespa orientalis in all of the 10 apiaries which are located in Avellino and Benevento provinces. This, we think it's mainly related to the fact that Naples, Caserta, and Salerno are super cities with a really high amount of anthropization, where actually Vespa orientalis can find not only food resources, because it is also a scavenger, but it can also have sites where it can actually build nests, which she feels comfortable with. We also monitored the loss of colonies, and what we found that in Naples, we lost nearly 40% of the colonies in different apiaries, in Caserta 20%. In the other provinces, luckily there were no loss of colonies. Obviously in Avellino and Benevento because Vespa orientalis was not there. Altogether, the beekeepers lamented a reduction in the production of the, like let's say an average reduction of 30%. Talking about associations, we had really, really bad results. We would have expected it, as it is actually their problem, they would actually answer it better. So we sent out 1,000 questionnaires. We only got 70 questionnaires coming in, 70 answers. And it was just because I actually went there knocking door to door. We also started as a citizen science project, an app, which is basically really simple to use. It is now on an ongoing internal trial. It works, unfortunately, till now only on Android. And basically, it gives the possibility to just give site links. And it is connected to a map, which works as a heating map. So we hope that actually once we actually put it on the market, it will be well used by citizens. Regarding the fight, what we wanted to test was the use of the most famous trap, which is actually used for hornets. So I'm talking about the tap trap. I actually was expecting this kind of results in the sense that it works quite well for Vespa glabro. We use different baits and quite a big amount of areas. We use 13 apiaries. In each apiary, we had at least 3 sets of bottles, and each set was composed of 3 bottles. Inside one bottle, we had basically what was recommended by the manufacturer, so beer and sugar. Then we had cat food in another one, and then we had peach with peach syrup. And basically, as you can see from the results, it worked really well with Vespa glabro. So actually, the manufacturer says that it works with hornets, but it is specific for Vespa glabro. Did not absolutely work for Vespa orientalis. In the same apiaries, we had a different kind of traps. They worked quite well. As you can see here, it is exactly the same bait that we use as a protein source in the bottles. They didn't like the bottles, they liked plates, and so different kind of nests. Considering also, let's say, loss of biodiversity, which was actually connected to the use of bottles, which is not something which is really specific. What we got, probably cannot see it here, it is the green fly. So I'm not an entomologist, I guess it is Lucilia serrata, something like that. And obviously we attracted other kind of predators, so cats were very, very interested, obviously, in cat food. We also built a team of people which were actually going and removing nests as soon as they rang us or they got in contact with us. And here you can see what I was talking about. So it is actually in the shutter box of a house. And you can see as you have to actually operate from inside the house, otherwise you risk to fall down from a balcony. Here it is where the heating system is on, so the boiler is on, and this obviously creates problems in using some kind of insecticides, pesticides, and substances which actually could be inflammables. And this is one of the primary nests that we actually found, which was actually in the shutter box here. And altogether, in let's say this year, we managed to remove actually 6 nests. This is something that I really think, like conversely to what some of you were saying, this is a fight between Vespa glabro and Vespa orientalis. We see this quite a bit. So Vespa glabro is far bigger than Vespa orientalis. And we see this quite frequently. So actually it manages to compete somehow and kill it and take it away from the apiary. Although the problems that we are seeing now in the past years is that actually we do not see Vespa glabro anymore. So we don't know if actually it is going to work in the future as a competitive, which actually is going to at least reduce or control the amount of Vespa orientalis that we have. We asked the beekeepers to keep track, so our beekeepers was actually working with us in the monitoring projects to give us some news about what was happening. And what we saw that the first The first sites started in March 2023, then they stopped. So they didn't see Vespa orientalis anymore, and they came back in June 2023. This is probably due to adverse climatic conditions. So we had a drop in temperatures, it started raining a lot. We found that the peak compared to 2022 was shifted for a month later, but the number of the animals is far higher. So it started later, but somehow they managed to produce and to get all together the animals out. We saw that the main activity of these animals, also if we think about their behavior and their ability to collect solar energy in their solar panels, it is mainly between 11 a.m. and 3 p.m. And we actually did not have any sightings above the sea level of 400 meters. On the other side, what kind of studies? Obviously, I'm a veterinary pathologist, and so I'm also, taking science under this perspective. So we actually found a detection of honeybee pathogens, particularly viruses. We actually have a soon-to-be print paper in which we actually found Trypanosomatidis and Nosema. We found a new parasite of not of Vespa orientalis, but it's a capillarides, so probably it is connected as, let's say, Vespa orientalis can be an intermediate host for some kind of capillarides species. And we now have ongoing studies which we are trying to use entomopathogenic agents to control this species. Considering the dissemination part, we do loads of activities with children, schools, different kind of populations, sectors. We also participate to beekeeping associations, although they're not really helpful when then we need their help. We have started and administrating a survey to people, and answers were great. So we actually managed to administer 300 surveys, 300 questionnaires, and we actually got back 300 questionnaires. So the question was structured having a first part on basic knowledge about entomology and particularly the beekeeping world. And we had 70% of actually correct. So we know that people are interested in this topic and they know quite about it. 59% of those who actually had correct bee knowledge were able to identify a hornet. So they could actually distinguish if it was a hornet, if it was a bee, or if it was Xylotopa. Among these ones, 38% actually could also identify Vespa orientalis. And this basically was the basis to try and understand, in order to get help from citizens, what kind of information we should actually give them to improve their knowledge and build, let's say, a whole bunch of new scientists. The conclusions, Vespa orientalis actually represents a great, huge, big problem, at least for southern Italy to now. We have no data on what actually is happening with wild bees. We talk a lot about beekeeping because obviously that is where we have the actual economic damage, but we have absolutely no studies in if it's actually predating wild bees or any other kind of insects. We need more studies on the species and especially on its adaptations, what's happening, why it is increasing. Is it climate change? Is it because of lack of food they actually found? Is it because we are basically raising honeybees which are stupid, not so anger against beekeepers, and so they're not so reactive to predators? We have to find a way of contacting beekeepers and beekeeper association because it's their problem, but they are also those who are actually in the field. We can actually get loads of information for them. The population is highly interested, so they are a great source of information if the information is somehow analyzed and if the people are actually educated to the topic. And obviously economic resources, at least in our regions, need to be allocated. So 15,000 euros is probably not what we actually need. We would need to have more money. I am a pathologist, so I can do my work, but obviously to fulfill all of these questions, definitely there is the need for each one of us to give its own expertise and its own competencies on the topic in order to fight altogether this problem, which is now the problem of southern Italy, but I guess probably is going to become a problem of all Italy and Europe. Thanks a lot. Sorry.
I'm very brilliant talk.
Thank you.
So I wanted to ask you, do you have an idea when the native species become a problem? I mean, since it was always present in campanias and conferences.
Well, this is something also that I would like to discuss. I mean, like, I'm not so expert in, you know, giving labels to stuff. So native, it means like I have had no reports until it became a problem. And this occurred, so I started my PhD in 2016. It occurred around 2019 in the Campania region. But this thing was going far, far, far already behind in Sicily, as an example.
I mean, I was in May 2012, and it was full of Vespa and Vespa. And I'm originally from Sicily, and I'm in the Iberville region, where it's like the most humid one. And when I was a child, I mean, it was full of trouble, and you had oriental hornets in Palermo, and now it's full of oriental hornets. So that is what my second question was: is it possible that you can link the increase in the population, like so the trouble that the Vespa is causing with, for example, climate change, so climatic parameters, because of course the species loss there is increasing and is also heading north. So it would be very interesting to go back to the point when the species started to be a problem for the beekeepers and beekeeping activities.
So to my knowledge, I would say around 2019, 2019. I think this connects to climate change, but I don't know if it is directly to climatic events, or if it is connected to the loss of biodiversity or something like that changing. Definitely what this year we saw is that it actually started the life cycle, then it stopped, and then it started again once the climatic conditions were favorable. It is strictly connected to the sun exposure. So if you have a rainy day like this, you will not see Vespa orientalis. It is not something which actually you find in the fields or in the wild. It is something that you find inside the city towns. And it's strictly connected. So in that case, I would say it finds loads of food. So you can always see it next to where you have, I don't know if you have here, cat colonies. So where people like old ladies go actually and feed the animals. You see it where they are, you have big piles of rubbish. And we are solving that problem in Naples, but it's still there. I know that in Rome, it's still a big problem. So this is something that actually, it is easy to reach food. So I think it's connected to the two things. Obviously, it would be super interesting to try to understand what is its main food and see if it is missing or not in that particular area. The problem is that the papers that we have and the studies that we have actually on the ecological and behavior or whatever things, they are dated to 1960s, 1970s. So, and they are, let's say, studies made where I would say it is actually native. So it is from Ishai, so we're talking about Turkey, Tunisia, Egypt. So, you know, has it changed his habits when it has arrived here in Italy? And what's happening in the environment to justify the fact that actually is raising in numbers and spreading across at least Italy?
I asked about the interaction between crabber and orientalis. Could you just repeat? So you did see a fighting situation, and what effect do you think?
Okay, so it is something that we see. It's not super frequent, but it's something that we actually see. So we did not see it once. I would say that I saw it around probably 15, 20 times in different places. So at least we know that there is, what we understood from that is that there is actually some sort of competition. The problem is that I haven't seen crabro anymore. So I actually had a friend that Federico knows. She asked me, so she wanted to come and do some sampling, and I said, okay, I haven't seen it, but I'll follow up with you and I'll tell you as soon as I see it. And I said, listen, I cannot help you giving you the samples. You need to go somewhere else because I'm not seeing it. But in the cases in which we see it, very often they actually fight. And as it is bigger, generally it manages to collect and then take away. Crabber manages, yes. But till now, I would not say that actually is effective way of controlling it.
There's a question from remote. Guess who? Please guess who? So he's saying, out of curiosity, does anybody know whether there is an interaction between small hive beetle and Vespa orientalis? For example, Calabria, Sicily, or elsewhere, additive, subtractive impact on bee colonies. So I guess it's for you and for the whole audience.
So luckily we do not have small hive beetle in Campania, and I don't have data on that. I don't think that people are actually studying this kind of thing.
I'm not aware.
I don't know if Professor Mutinelli knows something about it. I guess they focus on one problem at a time. I mean, like if they have the small hive in Pisa, that's fair enough of a problem for them.
Last question, yeah.
After seeing your picture from a comb of Vespa orientalis, I have two things to think of. Do you know the width of the cell? How big is a cell? And how what's the size of the hornets in comparison with the crabro and the velutina?
Okay, so I cannot give you actually, numbers. I could say approximately. So I would say that regarding the primary nests, so I would say it is about 1 centimeter diameter for Vespa orientalis. So are you talking about the cells?
The cells, because when we are looking at an empty nest of German wasp or something like that, they always say, use a pencil of the IKEA.
No, that's not. That's
smaller than
that. I would say is a pencil and a half at least.
Oh, so they are really big because velutina, it just fits in every cell.
No.
And from the German wasp, it's only 4 millimeters. I think from the velutina, it's 7 millimeters.
No, I'm talking about I think around 8 millimeters to 1 centimeters, probably. Something like that. So it's like it's big. It's big, and the animals are big. So like Vespa, crabro would be around. Also, I measured a sample which was 6 centimeters. So like you have a full beast like this, and but it ranges around 5 centimeters, I would say. While Vespa orientalis, it is a little bit smaller, so it would be around, I would say, 4 centimeters. Yeah, but probably entomologists can give you the actual answer, like perfect measuring about it.
Anybody else? Last chance. Federico? Okay, thank you very much.